Bismillahirrahmanirrahim. Dear students, Assalamu alaikum. This is a lecture about ribosomes. The ribosomes, they are the small, dense. The ribosomes are small and dense, rounded, and granular particles. And granular particles of the ribonucleoprotein. So the ribosomes, the nature of ribosomes are that they are the ribonucleoprotein, that it contains the ribosomal RNA and plus protein. Now where the ribosomes are present, so the ribosomes they are present in prokaryotes as well as in eukaryotes. In prokaryotic cell, the ribosomes, they are present freely in cytoplasm, while in eukaryotic cell, the ribosome present freely in cytoplasm as well as it is present in bounded form with endoplasmic reticulum and nuclear membrane. The outer nuclear membrane. The nuclear membrane is a double membrane, the outer and the inner. So the outer membrane also contain the ribosomes. Now the ribosomes in eukaryotes, they are also present in the matrix of mitochondria. They are also present in the matrix of mitochondria, and in plants they are also present in chloroplast. Now the ribosomes, there is a long history of the ribosomes, but simply we can say that it was discovered by Pallet in 1955. Now all the cells contain ribosomes. All the cells contain ribosomes. But those cells which are involved in ectoprotein synthesis, so they will contain more number of ribosomes. Now how we can isolate the ribosomes, the method of isolation, that how we can isolate ribosomes, the method of isolation. The ribosomes, it can be isolated by centrifugation method. Centrifugation is a method in which the cellular particles isolated or separated from each other by centrifuge. The centrifuge applies centrifugal forces which can separate the molecules from each other. The unit of ribosome is Swedberg, S. So S stand for Swedberg. Now this S of the Swedberg unit, it is related, the S is related with the size and molecular weight. It is related with the size and molecular weight of in molecular weight of ribosome. For example, we say that the ribosome, it may be 70S or it may be 80S. So this S, it represents the size and the molecular weight of the ribosomal subunit. Now the types of ribosome. Types of ribosome. So there are majorly two types of ribosomes. Majorly there are two types of ribosomes. The 70S ribosome. The 70S ribosome, it is present in the prokaryotic cell. For example, in bacterial cell. So the bacteria, it contain the 70S ribosome. Now the 70S ribosome further, it consists of two subunits. The 70S, it consists of the 30S unit subunit and the 50S subunit. Now the 30S subunit further, it contains the 16S subunit of ribosomal RNA and it contains 21 types of proteins. Now the 50S ribosomal subunit, it consists of 23S subunit and 5S subunit. These are the ribosomal RNA subunits and it contains 34 proteins. So we can say that 55 proteins are involved in the formation of 70S ribosomes in the prokaryotic cell. The 70S ribosome, it is also present chloroplast. Now in eukaryotes, the ribosome is 80S. The 80S further, it consists of two subunits, the 60S subunit and the 40S subunit. The 60S subunit, it consists of 28S subunit, 28S. The other is 5.8S and 5S ribosomal RNA, and it contains 40 proteins. 40 proteins. And the 40S subunit, it consists of 18S ribosomal RNA subunit and it contains 30 proteins. Now the mitochondria. The mitochondria, it contains 55S ribosomal RNA. The 55S further, it consists of two subunits. It also contains two subunits. The two subunits are the 39S ribosomal RNA and the other one is the 28S. The 39S further, it consists of the 21S ribosomal RNA and the 28S, it consists of 20S ribosomal RNA. The number of proteins I don't know, but it also contains proteins because the ribosome is the ribonucleoproteins. Now the number of ribosomes. The number of ribosomes, it is different in prokaryotes as well as in the eukaryotes. For example, E. coli. The E. coli, it contains about 10,000 ribosomes. In each E. coli there are present 10,000 ribosomes and it forms the 25% of the total mass of cell. the total mass of cell. In eukaryotes, in mammalian cell, the ribosomes are the 10 million ribosomes per cell. 10 million ribosomes per cell. Now the each of the ribosome, that is also about twice as large as prokaryotic ribosomes. Each is twice in size as compared to the prokaryotic ribosomes. So this is about the number of ribosomes. Now the structure of ribosomes. The structure of ribosomes. Structure of ribosomes. The ribosomes, as you know that it contains the two subunits, the larger subunit and the smaller subunits. The two subunits. The two subunits are the larger and the smaller. The larger and smaller. These are the two subunits. The ribosomes, they are the spheroid structure. It is about the ribosomes are spheroid structure, and it is about 150 to 250 angstrom in diameter. Now the ribosome, as it contains the two subunits, so these two subunits they are freely present in prokaryotes. In prokaryotes, they are present freely in cytoplasm. While in eukaryotes, the larger subunit, it is attached to the membrane of endoplasmic reticulum, or it may be to the nuclear membrane. The larger subunit is attached to the membrane, while the smaller is present freely. The smaller is freely. And the two subunits in isolated form, they are called monosome. They are called monosomes. The larger subunit, it has a dome shape. The larger subunit, it has a dome shaped, and the smaller is like a cap on it. For example, this is the larger 60s, and the smaller subunit, it is just like a cap on it. So these are the two subunits. Now the two subunits, how they are attached with each other. So during protein synthesis, the two subunits are attached. The two subunits are attached with the help of magnesium ions. With the help of magnesium ions. So how much magnesium ions are required? So magnesium ions 0.001 mole. Magnesium ions are required for the attachment of the two subunits. And when the two subunits they attach, when the monosomes attach with each other, so it form dimer. For example, this is the smaller subunit, this is the larger, so they are separated, so this is monosome, this is also monosome. Now when they attach with each other, now this is called dimer. So during protein synthesis, the two monosomes which are separated from each other, they will be aggregated with each other for protein synthesis. The main role of ribosome is protein synthesis. So this is about today's lecture, and the rest of about the ribosome, it will be discussed in the upcoming lectures. Thanks.